This is question 2 of the core Key Assessment 3 paper 1. We have got influenza, the flu, and we have got, it says it's a virus. Okay, so we're interested in viruses. I'm thinking of cell structure topic now. And it's a virus, it's a protein capsid which surrounds the enzyme RNA polymerase and its genetic material. So we've got some genetic material inside, and it said we've got an enzyme and we've got a capsid around that. So some sort of outside. Okay. That is made of protein, and the RNA is a nucleic acid. Okay, so that's the structure of the virus. Excellent diagram, I know. Describe the differences between the genetic material of the virus and the genetic material found in the cell. So remember, this is an animal cell in the nose. You might have a nucleus, mitochondria and so on, ribosomes, endoplasmic reticulum, Golgis. And in this nucleus you've got the genetic material. These are in chromosomes, but if you zoomed in and looked at the structure of the genetic material, it would look, it would be that sort of structure. And that would be DNA rather than RNA. So this question is really, do you know the difference between the DNA and the RNA? So you're just going to your table and going, well, if I give you one, the DNA is double-stranded, whereas the RNA is single-stranded, and answers like that. So that's fairly straightforward. Then we've got a lovely diagram showing how the virus infects the cell and replicates more viruses. And that's how they work. And these cells eventually that are the old cell that's been taken over and lysed, that's what snot is, largely. Right, state the name of the process occurring between stages 1 and 2. So between 1 and 2, what's happening? So I can see this cell is, this virus is on the edge of the cell, and now it seems to be inside the cell. So something going through the membrane, but it's too big to go through channels or proteins or anything like that. We have got the membrane enveloping the virus and forming a kind of vesicle with it in, it's going through that way, engulfing, maybe a vesicle, who knows. Looks like it's got a vesicle. So what's that method of transport called? That's so the one where it's going in. And there's a couple of, there's an overall sort of title, or you could mention the fact that it's solid going in. Stage 3. So stage 3, viral RNA enters the nucleus, and it is replicated. The RNA is replicated by viral RNA polymerase. Right. The capsid is broken down. RNA is replicated. Apply your knowledge of RNA polymerase. Right. Apply your knowledge of RNA polymerase to describe how the viral RNA is replicated. What do we know? Where have we met it before? RNA polymerase. We've met it in protein synthesis. So we're just really talking about our knowledge of transcription, that sort of knowledge. So we are talking about templates, adding nucleotides, which nucleotides we're adding, what the enzyme is actually doing. So similar to our transcription. To complete the replication of the virus, capsids need to be produced. Describe how the proteins in the capsid are produced. So the key thing here is describe how the proteins are produced. Protein synthesis. So protein synthesis. Where have we met that before? In protein synthesis. And it's the second part of it. So making viral proteins. So we're going to talk, it's similar to translation. So we're going to talk about all the translation things. You know, ribosomes and what's going on there. So just dump your knowledge on that for 5 marks. Short, snappy points. Make sure you say at least 5 things and you should be okay. And the last question, explain why the virus is unable to synthesize its own capsid. Well, we just mentioned it, you need a ribosome to make a protein, and then you need to sort of explain why that item there can't on its own produce a protein. It doesn't have the necessary equipment to do it, and you just need to put that into words. And that is 16 marks, of which a lot of it was just your own knowledge.